And I would like to thank everyone for coming out to hear me talk about weeds for the next hour. As JD said, I'm going to be talking about herbicide control options, looking at Palmer amaranth and some water hemp. So to give some background on these species, for those of you not as familiar, Palmer amaranth is an introduced species to Kentucky. It is native to Mexico in the far southwest regions of the United States. Water hemp is a very close relative of Palmer amaranth, and it is native to the Great Plains region of the United States. And neither of these species were a major player in agriculture in Kentucky or in much of the eastern U.S. until recent years. So in 2010, some surveys were conducted through the Cooperative Extension Service that polled local county extension agents. And that survey determined that there are 9 counties in the state that either had one or both of these species present. And you'll see they're clustered along the Ohio and Mississippi rivers in the far western regions of the state. But we fast forward here to 2017 today, and we now have 41 counties that have confirmed populations of water hemp. For Palmer amaranth today, over half the counties in the state now have confirmed populations of Palmer amaranth. And I'd venture to guess some of these counties that aren't shaded, it's probably there. It just hasn't been found and identified yet. So we get a feel for how quickly these species can spread from one geographic region to another. And so I want to mention some of the ways that they spread and their sources of introduction. And the most common method of introduction we've seen for these species is through harvest equipment, particularly the purchase and movement of used harvest equipment, such as combines and combine heads. Another common method of introduction we see is through the use of cottonseed hulls is a livestock feed additive. That's pretty common in dairy production. And Palmer amaranth is the most common and most troublesome weed species plaguing cotton production in the South. So it's a safe bet that when cottonseed hulls are being moved around, there's likely some Palmer amaranth or water hemp seed with it. And you can see the seed over here, these black dots to the right of this penny. That's Palmer or water hemp seed. It's about the size of a pinhead. So it can very easily get caught up in the inner workings of machinery, plant material, things of that nature to be spread. So another common method of introduction that we've seen, particularly here in central Kentucky, is through the planting of uncertified cover crop seed. So we all know when you buy certified seed, it's going to come with a weed-free guarantee. But we've seen some producers here in central Kentucky, in an effort to save some money, have instead planted uncertified seed that doesn't come with that weed-free guarantee, and they have introduced these species to their fields inadvertently in that way. And that's actually the method of introduction for one of my experiment sites. And then the most interesting method of introduction that I've seen talked about in some of our bordering states is through the digestive tract of migratory birds such as ducks and geese. And there's been some work shown that they have the potential to transport this seed up to several hundred miles from the point of ingestion. And so a 2016 Weed Science Society of America survey concluded that Palmer amaranth and water hemp are the #1 and 4th most troublesome weed species in the United States today. And much of that troublesome nature can be traced back to the biology of these species. And so both Palmer amaranth and water hemp are summer annuals. They're also what's known as dioecious, meaning male and female flowers occur on separate plants. So you have gendered plants, if you will. And so anytime these species are reproducing, there's a crossing of genetic material from multiple parent plants. When you combine that with the prolific seed producing capabilities of up to 500,000 seed per female plant, and some of that varies. I've seen estimates as much as 1 million, or down to 400,000 under optimal conditions. it gives these two species the ability to rapidly develop resistance to single and multiple sites of herbicide activity. And so I'm going to talk a lot about herbicide sites of activity, and so I want to define that for those in the audience not as familiar with that term. And so a site of herbicide activity is essentially the biochemical pathway or the biologic function within a plant that a herbicide targets. So different herbicides act differently in the plant to control it. And so when the herbicide molecules are taken up by a plant, they're going to move around in the plant, find what we call a target site that they're going to bind to interrupt a biologic function. And we classify these. We have a naming and numbering system that we classify group similar or like herbicide sites of activity together. And I also want to define for you herbicide resistance. What is it and how does it develop? I'm going to talk a lot about that today too. And so herbicide resistance can be defined as the inherited ability of a plant to survive and reproduce following what would typically be a lethal herbicide dose. And so most herbicide resistance we see is the result of what we call a target site mutation. And it's essentially a beneficial random mutation that occurs some point during reproduction that changes that target site that the herbicide molecules would go and bind to. And it prevents them from being able to bind. And so it prevents them from being able to interrupt those biological functions. And so this diagram is going to kind of help explain, if we have one or two plants within a field that have inherited that genetic mutation that allows them to withstand a given herbicide application, if that herbicide's applied alone, that plant's going to have the potential to survive it, reproduce, and then it will spread progeny that inherits that resistance trait. And over time, if we're not rotating our different herbicide sites of action, we're applying the same herbicides over and over, eventually the dynamics of that population, the proportion is going to shift to greater herbicide resistance, as we see in this diagram here. And so I also want to give you an update, kind of an overview of the state of herbicide resistance for these two species across the U.S. And there's currently 27 states in the U.S. that have glyphosate-resistant Palmer amaranth populations. And glyphosate is the active ingredient in Roundup. 13 of those states have Palmer amaranth populations with multiple herbicide resistance. That's stacked resistance to multiple sites of herbicide activity. For Waterhemp, it's a little less widespread, only 19 states with glyphosate-resistant populations, and 7 of which that contain multiple herbicide resistance. And as these species have spread, as we saw those maps across the state, and herbicide resistance has spread with them, we started to see some more fields that look like this. And this was taken in 2015 at one of my experiment sites the year before we began working there. And I believe it was last year I drove through central Illinois, and I saw a lot of similar fields where you could see these inflorescences sticking up out of the crop canopy. And when we have the lack of control that we see here, and we have this many plants, per square meter competing with our crops, we can see some pretty drastic yield losses resulting from competition with these species, and in particular Palmer amaranth, where we've seen as much as 91% yield loss in corn and 78% yield loss in soybeans. And that's simply economically unsustainable for our crop production systems. So now that I have stage set, I want to set in on my field trials. And so I'm going to talk about the 2 different years separately due to some variations in the experimental design and the treatment list. I'm going to start with the first year, the 2016 field trial. And so these trials were conducted on a field site in far western Kentucky in Ballard County near the town of Barlow. It's about 30 minutes west of Paducah. And we first became aware of this site because the producer there began to believe he had some multiple resistant Palmer amaranth population. And he actually had a mixed population of Palmer amaranth and Waterhemp together, which we don't see that very often. Usually it's one or the other. And so he had had this field in continuous soybean production for about 5 or 6 years, and was beginning to see some lack of control in these fields. And so plant tissue samples were collected from this field. 10 of Waterhemp and 10 of Palmer Amaranth were sent to the University of Illinois for some genetic analysis for them to test for the presence of some of the suspected herbicide resistance. And they were able to confirm with us that 3 out of 10 Palmer Amaranth plants were tested positive for PPO inhibitor resistance, and all 10 of the Waterhemp samples tested positive for the PPO inhibitor resistance. So the experimental design was a randomized complete block design utilizing 3 replications. This was on a fallow field setting. We did not plant a crop. And the treatment included 32 total treatments. So it's a pretty lengthy treatment list. And those treatments varied in their application timing as well as the herbicide composition. So this is the first portion of the treatment list. These are soil-applied pre-emerged treatments. So these would be applied to the soil before emergence of the target weed species. And you'll see starting at the top with the atrazine and going down the list, we're going from single to multiple sites of herbicide activity. And so for our soil-applied pre's, it ranged from 1 to 3 sites of herbicide activity contained within each treatment. For the foliar-applied post-treatments, these went on later in the season after emergence of our Palmer and Waterhemp plants and targeted them when they were 3 to 4 inches tall. And this did not follow the soil-applied pre's. Post was the only treatments we were evaluating here. You'll see they range from 1 to 2 sites of herbicide activity. And then we put the two together in some pre-post combination treatments that received the soil-applied pre up front, followed by a later season post-application. And they range from 3 to 4 sites of herbicide activity in each treatment. And so I collected a few different types of data to determine treatment effectiveness. The first were some percent visual control ratings taken throughout the season. Also collected some plant height measurements, where 6 plants were chosen at random within each plot and measured for their height. And then we had plant density counts, where meter square quadrats were dropped within each plot, and the number of plants within that quadrat was counted. So one of the things that we really wanted to look at first and target is there a relationship with the number of sites of herbicide activity that we apply and the corresponding effectiveness of control? And what we found was increasing from single to multiple sites of herbicide activity led to an increase in control. And that each time we added an additional site of herbicide activity, as we can see in this table, we saw a significant increase in the level of control. So this picture here, this plot is a plot that received 4 sites of herbicide activity during this trial. And you can see there's not a living thing in it. It's bare soil. So we would be very comfortable in knowing that if we had a crop planted, it's not going to be susceptible to competition. Whereas on the edge of this plot, some of these adjacent plots that received less, only 1 to 2 sites of herbicide activity likely, we see a complete lack of control. And you can get a feel for just how dense the population that we're working with is from that picture as well. The density and height measurements reaffirm the visual control data, and that our treatments containing 3 to 4 sites of herbicide activity achieved greater control than those treatments containing only 1 or 2. So we had greater control ratings, we had shorter overall plant heights, and we had less plants per square meter in those treatments containing 3 to 4 sites of herbicide activity. I also ran some comparisons in SAS, comparing between these numbers of sites of activity contained within each treatment. The only comparison that was not significant is the 1 to 2. And this kind of tells me that the benefit of going from 1 to 2 sites of activity could be pretty minimal. But that going from 1 to 3 or 2 to 4 sites of activity we could see a much greater increase in the level of control seen.
I hate to interrupt. Quick question. Are these just average across all your sites of action?
Yes, sir.
One.
Yes, sir. All right, so now I want to talk about some specific herbicide treatments. And we're going to start with the single site of action soil applied pre's. So again, these went down before emergence of the Palmer and Waterhemp plants to the bare soil. So looking at our first rating date here, 24 days after application, we see several treatments that are providing pretty effective control, ranging from 85 to almost 100% control. But as we move farther across to the subsequent rating dates, we see several of those treatments begin to stumble, and the effectiveness of their treatment quickly tails off. And we can pick out specifically one treatment in particular, the Zidua, which is a group 15 long-chain fatty acid inhibitor that provides the most effective control across all three rating dates. Now looking up here at the top at the Atrazine and metribuzin, those are both group 5 photosystem 2 inhibitors. They failed to provide adequate control across any of the rating dates. And I think that's a little bit problematic because atrazine is one of the most commonly used herbicides in the world. It's heavily relied upon for our weed control in corn systems. And so trying to, rotating to a corn system, if you have these species present, you may see some issues, with control if you're relying on those atrazine treatments to provide the bulk of your control. Now looking at some comparisons between the soil-applied PPO inhibitors. Start here with the Valor-SX. That we saw that in the previous slide as a single site of action treatment that started out with pretty fair control at 85% in our first rating date, but that quickly falls off across the later rating dates. But in the case with the Fierce and Fierce XLT treatments, we've added in another site of herbicide activity, particularly the pyroxasulfone, which is the active ingredient in Zidua from the previous slide, that provided such effective control. So when we add those two together, we see a very large increase in the efficacy of control, especially at our latest rating date here, 42 days after application. We're achieving almost 60% control, compared to the 10 and 20% control provided by the prefix treatment and the Valor treatment alone. So I'm going to move to our foliar-applied post treatments. Again, these did not follow the pre. These were applied later in the season when the Palmer and Waterhemp plants averaged 3 to 4 inches in height. And with the exception of the glyphosate treatments, we really didn't see any statistical separation between all other post-herbicide treatments. And that's not surprising due to the known resistance to glyphosate in this population. And we assume that most of these populations of Palmer and Waterhemp are carrying glyphosate resistance with them. But what was surprising is this Flexstar treatment, or fomesafen, which is a PPO inhibitor, provided pretty fair control, equal to that of all the other post treatments. In knowing that there's PPO resistance in this population, I expected those numbers to be much smaller, more in line with the glyphosate treatments. And so that's telling me that in 2016, there was still a lot of segregation in this population. We had pockets of PPO resistance, but it was not yet widespread across the entire field. And we also see from this slide, moving from the 10 to 18 days after application rating, we see some pretty quick fall off in these treatments in just a week's time. That's likely due to the lack of residual control in most of these treatments. So they're, alone, just by themselves are going to provide pretty short-term control of these species. Putting the two together in our pre-post combination treatments, we had a soil-applied pre up front, followed by a later season post. The first two columns, these rating dates, are only analyzing the pre-herbicides. The post was applied after rating on the 16th date here. And so what really stands out to me is the increase in control when we compare the visual ratings with the pre-post combinations, the numbers are much higher than the pre's alone at 42 days. If you remember from a couple slides ago, those single site of action pre's were barely, averaging 10 to 20% across them at the 42-day after application rating date. Whereas when we combine the pre and post together, most of our treatments are averaging 80% control at 42 days after application. So now I want to move to the 2017 field trials. And I mentioned that these trials, we changed up the experiment design and treatment list in 2017. And so we maintained the Barlow site, but we added an additional trial site in Bourbon County, near the town of Paris, here in central Kentucky. And we kept the most effective treatments, pre and post treatments, from the 2016 field trial and carried them over into 2017 in an effort to really hone in and pick out the few most effective treatments that we could then go to Kentucky crop producers and extension agents and feel comfortable and confident in our recommendations to them. So we changed from the randomized complete block experimental design and instead employed a factorial design where I had 5 soil applied pre's that were the first factor, and then the second factor were my 6 post treatments, 5 of which were post herbicides, and there was a untreated post where we were only analyzing the soil applied pre for its season-long control. So we're familiar with the Barlow site, but I need to introduce the Paris site to you. And we first went to this site in 2016, and we were called out because the producer did not realize he had these 2 species present in this field. And it quickly got away from him, and the Palmer Amaranth actually overran the soybeans, and they were looking for recommendations on what to do next, where to go. And so this picture here is actually this producer bush hogging down about 30 acres of soybeans because the population was so dense and he had such a lack of control in this field that in an effort to prevent spread of this seed to new fields and reduce seed production in this field, we determined it was best just to destroy that crop and the weed species present. And the seed in this field very likely came from another field of his in the same county. And a few years ago, we first ran across this crop producer because he had some Palmer Amaranth. He wanted us to come out an idea and try and help him understand where it came from. Because in a matter of one growing season, he went from not having it to having a pretty good stand of it that was competing pretty heavily with the soybeans. And what we found was we're able to trace this seed back to some uncertified cover crop wheat seed he'd planted. I actually sifted through some leftover seed and was able to find fragments of the seed to determine that. And so again, we took the plant tissue samples, sent them to the University of Illinois, where they performed their genetic analysis to confirm or deny our suspicions on resistance. And so at the Barlow site, we have resistance to 3 sites of herbicide activity, ALS inhibitors, PPO inhibitors, and then the EPSPS is glyphosate. At the Paris site, we only have confirmed resistance there to 2 sites of herbicide activity. So here's my treatment list for 2017. The first thing you'll notice is it's much shorter than the 2016 trials. So the top half, those first 5 treatments are my soil applied pre's, and that made up the first factor for this study. The bottom half are the foliar-applied post that made up the second factor for this study. And you'll see that all these treatments, the pre's contain 2 to 3 sites of herbicide activity. We did that seeing the effectiveness with the increased diversity of herbicide sites of activity in 2016. In most of our post applications, with the exception of the Liberty by itself treatment, contain 2 sites of herbicide activity. So to determine treatment effectiveness, I continued the visual control ratings and also took the plant density counts where I counted the number of plants per 10 square meters. We also collected some plant biomass samples where plants were cut off at the ground. They were collected, dried, and then weighed for their biomass plant material weight. But I'm not going to be talking about those today due to some time constraints. So due to a lack of interaction between my two experimental sites, I was able to combine the soil-applied pre's together across the experiment sites. And so these first two columns, I have the visual control ratings for the pre's only 4 weeks after application listed here. Within this last column are the two sites pulled together. And we see that Fierce XLT and Intimidator provided the most effective control across both sites 4 weeks after application. Both, averaging well over 90% control. Whereas the Broad XXC treatment failed to provide adequate control here across both experiment sites. Same thing we're looking at here, except the only difference is this is ratings taken 2 weeks later or 6 weeks after treatment application. Again, pulled across sites, similar story. Our Fierce XLT and Intimidator treatments averaging over 80% control provided the most effective treatment, most effective control of these populations. The three other soil-applied pre-treatments, all three failed to provide 70% control. So I didn't have an interaction between my two factors. So I analyzed the pre and the post data separately. And for the post, there was an interaction between experiment sites. So I was not able to combine those across experimental locations. So this slide is showing us visual control ratings 8 and 20 days after post applications for the Barlow experiment site. And at my first rating date here, 8 days after the post application, there's a lot of variation between treatments and a lot of different statistical separation happening. But I want to focus your attention on the two treatments containing the herbicide Liberty. In both those treatments containing Liberty, Liberty is a contact herbicide, so it's going to work quickly. The symptomology from those applications is going to show up quickly, and it's going to kill the plants off quickly. And so I think we're seeing this these high control ratings, over 90%, only a week after application, due to the contact nature of these herbicides. Now, if we move to the later rating date there, 20 days after post-application, we've seen that many of the other treatments have now caught up and are providing comparable or exceeding the control provided by Liberty. And so my top two treatments, the Enlist Duo and the XtendiMax plus Roundup Power Max, both contain glyphosate and a Group 4 growth regulator herbicide. And both those are systemic herbicides. So when they're applied, they have to be taken up by the plant, translocated, and then they have to go and bind with their target site. And so they work slower. So it took them a little bit longer to achieve effective control, but we see a jump here in the ratings from the first to the second rating date. For the visual control ratings of post-emerge treatments at the parasite, with the exception of the untreated post-treatment, there was no statistical separation here for the first rating date 12 days after application. All treatments are averaging, at least 64% control. Moving to the second rating date here, it's a little later, 30 days after post. Now all treatments are failing to provide 50% control. And so I think there's a couple things at play why we're seeing such low control at this site. One goes back to the density of the population we're working with and the competitive nature. But as I said before, this is a fallow field setting, so I don't have a crop in there shading out the ground and competing with these weed species. So I think we're seeing a quicker fall off in the control ratings due to that. So I also took the plant density counts, number of plants per 10 square meters. And there was an interaction between experiment sites. So this is just the plant density counts for the Barlow site. And this is before the post application occurred. So we're only analyzing the pre's. But it tells a similar story as the visual control ratings, and that the Fierce XLT and Intimidator treatments are providing us with the most effective control of this population. Now moving to some plant density counts taken later in the season after post-application. So now we're looking at the post-treatments, and there was not an interaction between factors for this data set either, so I wasn't able to, I had to analyze the pre and post separately. And so with the exception of the untreated post, there's not much difference between all our herbicide treatments. We've got really low plant densities, only a few plants per square meter, but we're not seeing much statistical separation. Now, I had a little slip up, and I didn't get the plant density counts collected for the pre-application, the pre-treatments at Paris. So I only have the post treatments to analyze and discuss with you. But the first thing I want to point out is the density of this population compared to Barlow. The untreated post here, averaging 12 days after the post application, almost 450 plants per 10 square meters. That's an absolute carpet. And in large portions of this field, you could walk across and your boots would never touch the soil. And other than the untreated post, there's no statistical separation here at the first rating date, and we're not seeing much across this later rating date. And so these kind of tell me that we rely very heavily on the soil applied pre's for our weed control. They're doing the majority of the work, and if we don't start our season clean, it's going to be really hard to make that up with our post treatments. And so you didn't see any atrazine data in the 2017 trials. That was not included in the factorial experiment, but we did embed some plots that received a early season soil applied pre-application of atrazine. And so across all 3 site years, we saw very poor control from atrazine treatments. And so we began to expect that there was atrazine resistance in these populations. Now, it did not show up in the genetic analysis tables, if you remember. And so the genetic analysis they're performing is looking for a target site mutation that I discussed earlier. But there is another type of resistance we see less commonly that's called metabolic resistance. And that might be what's at play here. I did try to perform some greenhouse screening trials to look into this further. I had varying success with that, but I really wasn't able to recreate what we saw in the field in the greenhouse. So we're still not 100% sure what we're seeing here, but that would necessitate some further investigation to delve into exactly what's happening, leading to the lack of control seen from atrazine treatments. So now I want to pull out and discuss with you some of the main takeaways I've learned from this research, and some of the things I like to stress to crop producers and extension agents when I'm discussing this work with them. And the first is that our chemical control programs targeting palmer amaranth or waterhemp should contain or start with a good soil applied pre that's going to contain at least 2 sites of herbicide activity. 3 is even better if we can get there. As I said, we're really heavily reliant, in my opinion, on our pre-herbicides for control of these species. I think they carry the brunt of the work. If we don't start clean, we're going to have a really tough time ending clean. So we're going to follow those soil applied pre's up with a timely applied post-application. We really want to target our post-application for these species when they're 3 inches or less. When they get above 3 inches, it becomes increasingly difficult to adequately control them. And I do recommend post-applications that contain 2 sites of herbicide activity. In particular, I like to see herbicide active ingredient thrown in that's going to give us some residual activity to help our crop get to canopy closure and compete better with these species. For our soil applied treatments, I do highly recommend combining group 14 PPO inhibitors with a group 15 long chain fatty acid inhibitor. Those were shown to be overall very effective across all 3 site years. And there's been some work shown in the literature that there's some additive effects when you combine these 2 sites of herbicide activity together. When choosing our group 15 herbicides, they're not all created equal. The pyroxasulfone, or the active ingredient in Zidua, was proven to be the most effective group 15 herbicide for control of palm amaranth and waterhemp. The acetochlor, or the active ingredient in Warrant, by itself proven to be very ineffective, provided very poor control of these species. But it could be used in combination with several other herbicides to get a very well-rounded, successful weed management program. And so, in Kentucky, in agriculture in general, we hear a lot of talk about these species. And there's a lot of worry with them, with their continued spread, their competitiveness, and then just the widespread herbicide resistance within them. And there's a lot of worry, especially in Kentucky, because it's such a new species to our state, and very few farmers have much experience, if any, dealing with these 2 species. And so there's a lot of worry around them. But I like to stress, when I'm discussing this with them, that my research shows we can still achieve very effective control of these species in our cropping systems today. Now, that's likely going to require some additional monetary investment up front in the form of increased chemical cost. But I think that's a very wise investment, because it's going to protect our crops from competition and yield losses due to lack of control in these species. And so that's going to wrap my presentation up. Before I open up for questions, I need to thank several people, starting with J.D. Green. Wouldn't be up here without him, so I've got to thank him. Got to thank my committee, Aaron Hermato and Travis Legleiter, for going through this process with me and helping me. And then there's several students, some of them are here, that have helped me in one way or another throughout this process, that have worked out in the field, where it's counting weeds or spraying plots or taking biomass samples, and they all had a hand in helping me get these trials out and performed, and bringing the data to you here today. So thanks everyone that helped me. And with that, I will open it up for any questions you may have. Yeah, Dr. Witt.
I'll start. Back to Atrazine. In your plot where you got very poor control, no control, you control other weeds?
Yes, but after saying that, the weed spectrum in these fields was dominated by palmer and waterhemp. They're so competitive that we didn't see many other weed species. They outcompeted everything else, but did see pretty good control of grasses and other weed species. It was mainly just the palmer and waterhemp that we had the lack of control with.
And related to Atrazine, tons of work done on other big weeds. With these herbicides you looked at, how does your control of either one of your food, it's fair to say smooth pigweed or red weed, has been in the literature forever? Do they get similar controls?
Oh,
Here's what I'm getting at. Is your acetochlor control with these 2 species similar to what you would see with smooth pigweed?
I would think you would see a little better control. As I go to North Central and I go to some of these meetings, that's interesting, because I've seen other states where they've had the opposite. The acetochlor has been the most effective group 15, whereas the Zidua has been the least effective. And so I'm not sure the complete relationship there.
Because that was, I'm you've seen a whole lot of acetochlor.
Sure.
Give excellent control of stuff.
Sure.
And better than the total, I think.
Yeah.
Across
and I saw that with other people's work. It's some of the meetings, but 3 years I saw the same thing happening. So I'm not sure if it's environmental soil type here in Kentucky that makes that relationship different. But there's definitely something at play there.
Did y'all take pH that follows like?
Yeah, it was right around 6.2, I believe. Yeah.
Not mention the formulation of acetylcholine.
Being encapsulated and it's taking longer to be released. Could be at play there with the quick emergence and quick growth of these species at such high photosynthetic rates. It's pretty easy for this species to quickly outgrow the desired range for herbicide applications.
One last question on the fly. On corn, unless you answer something I don't agree with. I don't get to do this anymore. On your corn materials, looks like Callisto is far and away the best. Did I see that correct?
Yes, sir. Yes, sir.
If you're going to tell a farmer to corn soybean rotation, what would you be using to keep that weed down in corn?
So a lot of people.
Favorite herbicide you've already said is worthless, so we'll go from there.
At least in Kentucky, or my work. So yeah, the Callisto was shown to be very effective. So HPPD inhibitors, although it didn't really work with those much, I think are very effective on these species. There's no known resistance to that in Kentucky. And actually at the parasite, it was surrounded in corn. And the producer there was using Armazon, another HPPD inhibitor. Armazon. And so I think the HPPD inhibitors could be very effective for this. Also, for our post control, our growth regulators, dicamba or status 2,4-D, those can be very effective at providing control. So I think up front, an HPPD inhibitor or something along the lines of a Lexar type herbicide where we're mixing in multiple sites herbicide activity. Maybe some atrazine, acetylchlor, mesotrione, and then following that up with some multiple sites post.
One of those big glyphosate.
For pre or post, it could be because glyphosate still has some value in our weed control systems. A lot of our grasses, Johnson grass and things, it still provides effective control of. It doesn't for our Amaranth species. But it still does have value in our weed control system. Any other questions? Yeah, Aaron.
Knowing absolutely nothing about the site.
I've got to wonder a couple things, and the pattern, if there isn't maybe a little bit of combine harvester pattern with scattering, maybe that this is new to this field and we're not seeing it completely widespread because we're seeing these long, parallel patterns. It could be something like that, or they could have sprayer issue. Maybe their sprayer isn't calibrated. Maybe they have some nozzles that are worn and don't have a good pattern or aren't applying the herbicides at the desired rate. That could also be at play there.
That was not one of your field sites.
No, this is not one of the field sites. The populations at my field sites were much overall more uniform than that, and didn't show these kind of patterns. So. Any other questions before we wrap it up? Sure, Dr. Witt.
How can somebody that's harvesting wheat, the difference in wheat seed size versus pigweed is enormous.
Sure.
How could they possibly be that poor at harvesting wheat get that seed in that bag?
You know, that's a good question. And their actual, methods in that being uncertified seed and not knowing the actual source, it is tough to answer. And I don't know if it was already caught up in the combine before they started harvesting wheat. Maybe the last thing they harvested before the wheat was, say, a soybean field that had this seed in it. And they didn't clean out their combine or combine head. And so maybe that seed that he had was some of the first, the first harvested, and that seed was left over from a previous crop or a previous field, would be my guess. Because theoretically, you think going through the combine and everything, going through the auger, offload to a truck and that it should, the Palmer and Waterhemp seed should shake out to the bottom.
So you said you saw pieces in that cover crop mix. Were they actually seeds or the little green?
It was the green kind of the seed covering there, yeah. My theory is that since the seed that I looked at was left over and it was taken out of his planter, that all the actual seed itself going through the field sifted down to the bottom of the planter boxes and got planted into the field. Well, if no one has any more questions, I'll wrap things up. And again, I want to thank everyone